Welcome to CureGNAO1 presentation of GNAO1 Mutant Gene Suppression Therapy. Do miracles happen? Are you waiting for a miracle? I don't know how to answer the question. I do know that you ask for a miracle, you look for a miracle, and if it doesn't happen, you roll up the sleeves and you make it happen. So that's the way I am envisioning this project. The reason being is that I realized long ago that no one's really going to help because nobody lives through what we are living. So to quite a few of the people that are involved or were involved, they don't understand the urgency, or if they do understand the urgency, it's not their child, so they don't live through what we live on every single day. So we have to push for it as much as we can, and it's up to us to make the miracle happen. Why did we create this nonprofit organization? The idea behind CureGNAO1 is to look for genetic therapies and treatments that can help our children as soon as possible. When I say this, it means that we are most likely not going to fund the research that is not directly applicable to getting the treatment. And so this way we can stay focused. We're probably not going to run conferences or have specific events. We want to be focused on getting the treatment and the help that our children need. I would like to present the CureGNAO1 parents team. Presently it is myself, Ilya Musayev, Igor Platonov from Moscow, Russia, and Stephanie Fernandez from Mexico. Our website is CureGNAO1.org. There's going to be a similar website for the Russian and Mexico sites. CureGNAO1.org is being worked on right now, and I'm hoping to complete it in the next couple of weeks. Why do we think mutant GNAO1 gene suppression therapy could be helpful? For me to answer that question, I need to first give you an overview of the science. One of the most common things you will hear in field of genetics is a term called haploinsufficiency. What is haploinsufficiency? Haploinsufficiency in genetics describes a model of dominant gene action in diploid organisms, in which a single copy of the standard, so-called wild type, allele at a locus in heterozygous combination with the variant allele is insufficient, that's the key word, to produce the standard phenotype. What in the world does this mean? Well, let me explain. So the first thing that you will notice that probably will confuse you is something called diploid organisms. Diploid organisms are organisms that have two alleles, one from each parent. In case of humans, in case of animals, insects, and others, for the most part, many organisms are diploid, where they will carry two alleles. In other words, two copies of your genetic code, one from each side or one from each parent. In case of humans and many other organisms, in many cases, you will see partial loss of function of specific gene. What that means is that in my picture there's allele 1 and allele 2. It could so happen because of genetic mutations that one of these two alleles is turned off. It could be present, but it's not going to produce any protein. There are many genes out there that can tolerate half of the allele, or in other words, just one of the two alleles functional. And they will produce 50% of the protein, or could possibly less, and that wouldn't affect the function of the body in any way. The question we're going to try to answer with this presentation is if GNAO1 can tolerate the silencing of one of the two alleles. In our case, it would be the mutant allele we would like to silence. The next thing we need to review is something called homozygous and heterozygous mutations. What is homozygous and heterozygous mutations? Heterozygous mutations are mutations that only happen in one of the two alleles. So for example, in case of GNAO1, most if not all of genetic errors are heterozygous. And that means that the error will only occur in one of the two alleles. Homozygous mutations are mutations that would occur in both of the alleles. In this picture, you will see two axes, which represents that both allele 1 and allele 2 has a specific error and specific gene. What we know about GNAO1 is that usually these mutations are not well tolerated. So if heterozygous mutations are somewhat tolerated, homozygous mutations are usually lethal. One of the most common ways researchers will look at haploinsufficiency and its effects is to create a genetically engineered mice where half of the gene is silenced. In other words, you have two alleles, one of them is turned off. In our case, there is already a mouse model for GNAO1 that has half of the gene silenced or one of the alleles turned off. And it's already documented. It's been studied by Dr. Nubik and by Dr. Martymyanov and other researchers. What's interesting about this model is that it describes a specific issues, and I'd like to read out what stands out the most. Mice homozygous for this target knockout have a decreased survival. 50% die at age of 7 or 7 weeks of age. In addition, there is an evidence of embryonic lethality. Mice exhibit a generalized tremor, impaired motor control, and increased sensitivity to pain, hyperactivity, and circling behavior. So I've highlighted the first word called homozygous, and that's very important because it means that the researchers were able to create mice with lack of GNAO1 or a complete silencing of GNAO1. Now let me explain why this is important in the next slides. So this picture has two mice, presumably male and female, and what's interesting about these mice is that they're both mutants in the sense that half of the GNAO1 is silenced. So the mouse on the left, you will see there are two alleles for two DNA strands, and you'll see an X there, and the same thing happens to the mouse on the right. What happens next is that you would cross these mice, and generally, if you remember something in biology courses, you would have three variations of mice when you cross two heterozygous mice. You will have wild type, which means they have two normal GNAO1 copies. I decided not to include them in this picture. And then you'll have homozygous pups and you'll have heterozygous pups. To better understand if GNAO1 can tolerate haploinsufficiency, we need to observe how homozygous pups and heterozygous pups live and survive. From what we know already, that in case of homozygous pups, the mutations are mostly lethal and they will have major abnormalities if they are not lethal initially. Whereas in heterozygous pups, which means one of the two alleles or half of GNAO1 is expressed, you will see some mild phenotypes. They're gender dependent and they're also mouse strain dependent, which means overall it's fairly mild. Has anyone studied the GNAO1 knockout mice with only one GNAO1 allele present? So the answer is yes. Dr. Nubik has done a lot of research back in 2014. In addition, his G203R mice that he thought were G203R actually turned out to be a knockout mice because of some other error he didn't notice. But based on his research, we actually learned a lot. And it comes down to the following. The males on the B6 strain, these are the kind of mice that they have, black 6, they exhibit seizures only when they administer a specific medication called PTZ. In other words, if you don't administer any medications, they're absolutely normal. Also, males on B6 strain have differences, mild differences when it comes to stride length and grasping. That is, when they were put on the treadmill, they would not run as much in comparison to the wild type mice. Males on 129 strain, which is interesting, with the same knockout, actually didn't have any seizure phenotype. They were very much comparable to the wild type. So what this suggests is that between different strains of mice, there could be some compensation happening for the lack of GNA1. This could potentially also apply to people. What's intriguing also is that females on the black 6 strain are completely normal. There's no issues of any kind. No stride issues, no grasping issues, and no seizure issues, even when you administer a PTZ medication, which is a very good sign again. What was also interesting is that they didn't exhibit antisocial behavior in his tests. So that suggests that from development perspective, they're okay. So in conclusion, the GNA1 heterozygous mice are viable. They have mild impact, but that impact is gender and strain dependent. What about homozygous? Unlike heterozygous pups, homozygous pups are actually a lot more troublesome, which is in a way a good sign because you would expect to see some problems there. So from what we know so far, what's been published is that they just have overall issues. They die, half of them or most of them die by 7 weeks of age. Very few of them are born, which means they have embryonic lethality, so it's very hard to create a colony of these mice to study them. They also exhibit tremor or impaired motor control, increased sensitivity to pain and circling behavior, which is in mice generally means it's autistic. And then issues with motor control, motor behavior, pain perception, and calcium channel regulation. Now, what strikes me as interesting and also important is that the fact that you are able to have mice that are homozygous with 2 of GNA1 alleles turned off, that in itself is a very promising thing, because that means something we don't know, but it just suggests that there are other genes, within the brain or biology that could compensate for the lack of the GNA1 gene. So the hope is that by silencing the mutant GNA1 gene, one, we will have 50% of the good GNA1, and then, humans are a little more evolved than mice, and there could be some other compensatory gene behavior to help offset the lack of GNA1 if such thing happens or exists. So based on what we see so far from the existing studies, it appears that 50% of GNA1 expression is tolerable, and it's a good sign. That means that mice and humans could potentially live with GNA1 expressed at 50%. Now, what have we done besides that to see if we can validate our hypothesis? On 3 separate occasions, we've tried to create GNA1 G203R mice. We needed these mice to test our gene therapy projects to check on their effectiveness. What happened in our case is that in 3 separate occasions, in 3 different places by 3 different researchers, in this case Dr. Newbick from Michigan State University, Dr. Alexei Dakin from Institute of Gene Biology, Russian Academy of Science, Moscow, as well as Dr. Vladimir Katanaev from University of Switzerland, Geneva. They've all tried many times, but they've done many sessions, close to 30 sessions, probably more, where they would try to create these mice. It is done through something called CRISPR-Knockin, where they would create a construct and inject it into an embryo, which will then go in and modify the GNAO1 gene and insert that mutation. What happened in our case is that we would have number of mice born with G203R variant, but they wouldn't live beyond specific age, or they would die in utero, or for whatever other reason they'll be non-viable. So what this tells us is that we know that G203R variant, as well as many other GNAO variants, they're very harmful to animals, and potentially they're also very harmful to humans. Suppressing that toxic GNAO1 variant as well as other variants would probably be beneficial and make children or people closer to what the heterozygous mouse mutant might be. We've also invested into a conditional GNAO1 G203R knock-in mouse model. The reason why we decided to go this approach is that the CRISPR knock-in, the traditional CRISPR knock-in mice that we tried to create were not viable. And so another way that, another approach we could take to get the mouse model that we need is to create a mouse that will have that mutation present but silenced. So what that means is that a mouse could live a normal life without any issues. If you do a genetic test, it'll come up as non-mutant. At a point when you decide that you need that mutation to be expressed in that mouse, you could give it a drug called tamoxifen, and then within 3 to 5 days, that genetic region will become exposed and the mouse will become mutant. So that is an extremely powerful tool. What we also get with the conditional knock-in is that we can selectively decide by breeding different kind of mice that we want the mutation only to be expressed in the brain, or we want it only to be expressed in the lungs or digestive tract or heart. So an extremely powerful, extremely flexible tool that also guarantees that it's going to live until a specific point. Now we know enough about mice. What do we know about people that are out there that could potentially have a mutation in the GNAO1 gene and only have half of it expressed? Are there people like that? So we know that in our GNAO1 community we have, I'd say, probably at least 30 different variants, and they're all, from what I've seen so far, some sort of point mutation, deletion, or some other issue. But I haven't seen anyone yet in the GNAO1 Facebook community that has a stop codon or intron splice mutation that could prevent the creation of GNAO1. However, we do have many tools at our disposal when it comes to researching that. And so what I've done so far is that I went to multiple sites, and just so you understand the way this works, anytime you go when you do a genetic test, so in your case you probably went to a hospital, to a doctor, he ran through a test, your result came back and it said you had this specific mutation. What happens is the company that runs that genetic test for you, periodically they will update something called DBSNP or some other database to track the mutations. And while the information about the user is stripped and hidden, these sites will have a count of how many reported GNAO1 mutations of this kind has been reported. So what I've done in this case is that I decided to download, there's about 37,662 different mutations that are reported, and I've decided to download them all and look through them to see if I can find a single mutation that is pathogenic, which means it's harmful, and it's also similar to a loss of function, like partial loss of function, meaning what we're trying to achieve, which is we're trying to silence one of the two alleles. There are people like that only have one functional allele. Just to summarize what I've mentioned in the previous slide, I have surveyed the CureGNAO1 family Facebook group looking for anyone who has partial loss of function mutation. I couldn't find any. I've also done similar things with the genomic database websites like DBSNP, ClinVar, GNOMAD, and others, and I couldn't find anyone or anything that would suggest that the loss of one of the two alleles is pathogenic. What was more interesting is that I found multiple cases where you would have a stop codon, which is basically it's a genetic error that happens in the beginning, at the very early beginning of the genetic sequence, which in theory would basically create a very small, it's essentially almost as if you had a loss of function. You didn't create that protein. You only created very little of it. So it would, if you look at it objectively, you would have one full normal copy of CureGNAO1 in allele 1, and the allele 2 would have just a little bit, just like, I don't know, maybe like 30 or 20 base pairs. And those cases, and there are at least, I believe 4 of them, they were not reported as pathogenic. So it is a good sign. It looks like we're going in the right direction, and potentially suppressing the mutant gene is not it wouldn't cause haploinsufficiency. What are the risks of gene suppression therapy? Are there any risks that we need to be aware of? So right now there are no known counter indicators that suggest that haploinsufficiency is an issue with CureGNAO1. It looks like we're borderline, we're 50% good, and that's a promising sign. Also, there isn't anything out there, there's no one who's done gene suppression therapy for CureGNAO1 to tell us otherwise. Any therapy, any drug, any medication has a potential for unknown side effects, and we'll find out. We will do a very rigorous animal studies, and you can't get this done without FDA's oversight. The very first stage of clinical trials, phase 1, all FDA cares about is that you do no harm. And so they will go out of their way to make you do many, many different tests to prove that your drug isn't causing any problems. And the next part is risk-benefit analysis. So what do we want to do? Do we want to do nothing and treat them with medications that barely work? Or do we want to try something novel? And again, yes, there are risks involved with any medication, and because there's just uncertainty, right? But we do know that time is of the essence, and we need to treat this as soon as possible. Because again, the disease is neurodegenerative, which means every day a very, very, very small portion of the brain cells could die. So the sooner we start the treatment, the better. We do have, as I mentioned, the CureGNAO1 conditional knock-in mice that we could try this on, and we will, to best of our abilities, try this on. What could potentially happen is that by trying that, it could delay the entire project by 10 plus months. And there's also a there's a safe approach done. So when you design these studies or trials, human trials, you will come up with a protocol, and the first thing that FDA would want to see is that you will do something called dose escalation, right? You will start with the very minimal dose, and then you will wait 2-3 weeks, and you will observe the child, and if you see nothing happening, you will inject the second dose, and then a third dose, and then eventually you'll get to the maximum dose that you think is right. And then every 2 or 3 months, most likely three, you would do a booster shot. And so this kind of treatment, if it's done with antisense oligonucleotides, is something that would be administered between 3 to 4 times a year. I want to briefly talk about the research and development team. So these are the 5 people that you see right now on the list. I want you to know that there's actually many, many more people that you're not seeing. So these are the lab leads, right? And within every lab, you would have between 3 to 10, 15 people that are involved. And at some point in time, different people will come in and help us at different stage. So the first person I would like for you to meet is Dr. Timofey Zatsepin. He's an excellent antisense and RNAi developer. He's the one who helps us with creation of these small molecules that are targeting the CureGNAO1 mutant gene. The next person is Dr. Alexey Wolfson. He runs a company called Advirna out of Boston, Massachusetts. Also an excellent guy. He is helping us with the validation. Similar to Dr. Mariana Bardina, who is also involved in another project, but in this case, she is also helping us to validate the molecules that Timofey has created. The next person is Dr. Kirill Martymyanov. You probably know him or met him in one of the conferences. He is our go-to G-alpha-O expert researcher. He's helping us tremendously, as well as Dr. Timothy Yu. Dr. Timothy Yu is actually a hero of mine, per se. He was the first person who created a custom personalized genetic therapy for a child named Mila. And Mila was suffering from Baton's disease. And what was remarkable about what he has done is that he was able to create a genetic therapy for that child that was specific to her within a year. So from the moment he started to the moment they began the clinical trials, took him exactly a year. Now this was done in 2018, which until before 2018 was unheard of. So based on Dr. Yu's experience, we can now create this therapy in the expedited manner. He has worked a lot with FDA to create something that's called N of 1 treatment model. He is also our lead clinician, and he will be helping us with the clinical trials as well as the studies that we are running through to get FDA approval. So this is the last slide, and I want to say thank you for listening through this presentation. I want to conclude this presentation with a thought. The reason why I am doing what I'm doing, I believe the reason why Igor and Stephanie are doing what they're doing, is we'll live for our children. And if they suffer, we will suffer. I can't imagine a life of my child continuously suffering. I don't know, I just can't picture this life where it'll be so bad day in, day out. So we need a solution. And if we don't do anything, there's obviously going to be no solution. We have to do something. We have to fight for our children. And the reason why I'm doing is that it's because if, let's say, we've done all this and we failed, at the very least I want to be able to tell my child on his last day that I've done everything I could. This is the only way I can live with no regrets. Again, thank you. Please join our efforts. Help us with fundraising. Help us with awareness. It's only 3 of us right now who are paying for this entire thing. And chances are we are not going to be able to pull off. We are going to be short. And we do need your help. Again, join us. Let's make miracles together for our children.